Welcome to the nDesign webinar series. In this webinar I'm going to cover how to use nDesign to place an order for a standard code set. There are other webinars that cover the elements ordering process, as well as a quick start webinar that takes you very quickly through the main aspects of the site. So with that, let's start this webinar, and I'll assume you're on the nDesign webpage. The nDesign Gateway homepage contains information on the application's features, links to perform specific functions, and also FAQs to address commonly asked questions. What we're going to do here is actually demonstrate searching on genes. So once we've selected the search utility, we have a number of choices. So one common case might be where I have a list of genes I'm already interested in. To do this, I'm going to use the gene list search, and you paste in a list of genes from Excel or a similar text-based program. You click the search button and then you get your results. The first thing you'll notice is that any gene not found will be reported at the top. And you'll also notice that there are actually two tabs here, Genes and Panels. The Panels tab allows me to see how many of my genes are in our existing panels. So in this case, we can see that 18 out of 20 of the genes that I put in my gene list are in our inflammation panel, and I can either hover over or click to see which of the genes from my list are present in this panel. But in this case, what I actually want to do is to design a code set using the genes that I searched for. It's important to note that gene list search will return results for your terms on both the name as well as the alternate names. So GDNF3 was not on my list, but it's returned here because it has an alternate name of ATF2. So be sure to examine your results before submitting any order requests and remove probes as desired. I can further investigate each individual gene by clicking on the plus and expanding it. And here what I see is all the different probes available for that gene. And you'll notice that one of the probes is defined as standard and elements chemistry. I can use the gene viewer to figure out whether I want to use the nanostring optimal probe, which can be used for both the standard and elements chemistry, or alternate probes, which are available for nDesign for elements code sets only. If you do want to order a code set with elements only probes as a standard code set, then export the code set as I'll show later, and simply contact sales with the exported spreadsheet, letting them know that you would prefer a standard code set. Because this demonstration is focused on nanostring standard code sets, I'm going to select all optimal probes. And I can do that simply by clicking up here, selecting all results, and now you'll see that all the optimal probes have been selected. And so I can just click on Add Selected to a code set. In this case, I want to add them to a new code set that we will call the demonstration code set. And at this point, I have to choose whether I want standard or elements chemistry. So I'm going to choose standard, and then I'm going to add the probes to that code set. So now that I've started a gene list using some genes I'm interested in, I might want to explore what other genes I might want to order for my study. I'll start this by selecting the Browse Genes tab to browse based on the different classifications that nDesign provides. Now that I've added probes to create a code set, you can see at the top of the page that I'm working in my code set. So anything we add probes to now will add those probes to the demonstration code set. You'll also notice that we are in the human organism because that was the default I chose at the beginning. You can also work with mouse and rat, and it's important to note that you aren't limited to these organisms. We've worked with many other organisms. Anything that has sequence information you can work with. And if you wish to work with something other than mouse, human or rat, please contact your sales representative or sales@nanostring.com. So notice on the left-hand side of the page here, you can see the different classifications that we offer for you to be able to search on. So for instance, if I wish to add the genes from the JAT-STAT signaling pathway to my code set, I can search for them here in the KEGG pathways. And I can just select that pathway. Now we see a description on the right hand side of the pathway, to which we can also jump to the KEGG web page and see more details on that whole pathway. Or we can look at the actual genes in the set, and we can now add them to our code set. So at the moment our code set has 21 genes. You'll notice if I add all of these that we now have 170 of the total of 800 we can put into a standard code set. So as well as adding directly from a pathway or a list of genes, I can also search on more generic terms. So for instance, I can search on the term growth hormone, and this is a Google-like search, so you need to put the words in quotes if you wish them to be searched together. And now what we're seeing is all the genes that have growth hormone in them, the pathways of the growth hormone, the gene ontology, and the KEGG disease. So we can choose anything we want, just as we would with anything else. Notice that some of these are already recorded as being in our code set. So for instance, I might want to add this gene to the code set, and I've now increased my code set by 1. I've now compiled a code set by browsing on classification, searching on keywords, and adding genes from my own list. So let's say I'm now happy with this code set and I want to proceed to submit my order request. So now I can go to my code set details page by selecting code set details up here. This page provides me with a list of all contents from my code set. I can remove a probe or add a unique custom probe name here using the edit link. Before proceeding with my order, I'd just like to point out the different functions and downloads available on the code set page, details page. So the new code set button allows you to copy the current code set to create a new code set. You can then add or remove probes as needed. This is not the way to do reorders where you don't want to make any changes, but obviously it's a good way of maintaining your previous code set whilst actually making changes to it. The share button allows you to send this code set to any other person that has a NanoString web login. This is helpful if you want to send the list to a collaborator, colleague, or similar. The code set details download is a spreadsheet of all of the probes and associated information as displayed on this page. The FASTA format exports the probe sequence information in the FASTA format, so you can use it for bioinformatics analysis outside of nDesign. Additionally, if you've already submitted your order request, reorder and cancel order will be available on this page. So once you've really reviewed your code set and these are the genes you want, you're ready to proceed. Finally, for every code set, there must be reference genes, often referred to as housekeeping genes, included. If you've already included your reference genes in the genes that you actually have placed in the code set, you can just select them here by saying yes. And this means that we'll know there are reference genes and that the electronic files that come with the system will be able to tell nSolver that that gene is a reference gene. If you don't have any reference genes in the set, we do have some commonly used reference genes that you can highlight here and select and place in your code set. We recommend a minimum of 3 reference genes in a code set, and obviously you can put as many as you wish in. So I could just put all of these in just to show you and add those to our code set. So my code set now has reference genes, and so I'm ready to proceed with my order request. So if we go back to the code set details, so I can now submit my order request. So to do this, you select submit for a quote and you put in the number of reactions that you'd like to use. There is a minimum of 48 reactions for a custom code set, and obviously you can go as high as you want. If this is a proof of principle experiment, which is a type of experiment that's run in-house at NanoString for evaluating the system, you would select yes here. But you should only select the proof of principle if you've already spoken to your sales team about doing a proof of principle experiment at NanoString. And also, if this panel is going to be run as a single cell assay, you should choose yes here. Even if only some of the reactions will be single cell, we need to know to actually do the single cell design. So when you click submit request, Submitting this is simply an order request that gets routed to the appropriate sales representative. They'll contact you to finalise your order. Likewise, as you can see here, I've received an order confirmation myself in the email, and for existing NanoStream customers that are familiar with the old way of ordering, the downstream sales process from here is basically unchanged. Thank you for listening to this webinar. I hope it proved useful and answered some of your questions about the nDesign Gateway. If you have any general nDesign questions, please contact webrequest@nanostream.com. If you have any questions regarding the Elements orders, please contact elements@nanostream.com and also contact sales@nanostream.com for purchase of any nCounter products. Thank you very much for listening, and please visit the website for other webinars on using the nDesign Gateway. Thank you very much.